Hey everyone, here's a little bit of information about cell reproduction. So let's look at cell reproduction in autosomal cells. That means cells that are not sexually reproducing cells. These are cells that are reproducing more of the same kind of cells. So here's the cycle at a glance, showing us all of the different types of phases: the interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Starting with interphase, there are three subphases. We have the G0, which is the non-dividing stage, G1, which is, or gap 1, which is between the end of mitosis and the S phase, and then we have the S phase, which is where DNA replicates, one chromosome becomes two sister chromatids, and then we also have the gap 2 where cells prepare for mitosis. Here's another look at interphase for you, where you can see the different phases leading into mitosis. Here we start with prophase. Characteristics that indicate the beginning of prophase are individual condensed chromosomes becoming visible, condensation continues through prophase, and the nucleolus disappears. You see that there's an assembling of the spindle apparatus, and then the microtubule apparatus is made of spindle fibers continues to assemble. There's a linking sister chromatids to the opposite poles. Showing you another glance at prophase here. And then we're moving on to metaphase. In metaphase, there's the division of the centromeres. The key characteristics of metaphase, and this is the stage when chromosomes align in the center of the cell, and then alignment occurs along the metaphase plate. This is not a physical structure, but it indicates where the future axis of the cell division occurs. Centromeres are equidistant from each pole. And we can see that here. The centromere splits in two, freeing the sister chromatids from one another, and all the centromeres divide in synchrony. And here's a live shot of metaphase for you. Moving on to anaphase. This is the shortest phase. This is during which the sister chromatids separate. The chromatids drawn to the pole which its kinetochore is attached, and separation is achieved by two simultaneous microtubular actions. The poles move apart, and the centromere moves toward the poles. And here's a live shot of anaphase for you. Moving on to telophase. This is where the separation of the chromatids completes partitioning of replicated genome. The spindle apparatus is disassembled, and the tubulin units of microtubules are used to build new cytoskeleton. There's a nuclear envelope reforms around each of the new chromosomes, and the chromosomes begin to uncoil to allow gene expression, and the nucleolus reappears. There's a live shot of telophase for you. Lastly, we have cytokinesis. Cytokinesis is when mitosis completes at the end of telophase. The replicated genome divided into two nuclei at opposite ends of the cell, and cytoplasmic organelles assort to regions that will become separated, and then the cleavage of the cell into two halves constitutes cytokinesis. The cell is pinched into two by a constructing belt of microfilaments, and the furrow deepens until the cell is pinched in two. Here's a live shot of cytokinesis. So to recap, we saw interphase, prophase, metaphase, anaphase, telophase, and cytokinesis to complete mitosis. Let's move on to look at cell reproduction in somatic cells. So do you ever wonder why we look like our parents? Each parent donates genes to their offspring via sexual reproduction. The genes combine to give different but similar looking offspring. Humans have 46 chromosomes, which consist of 23 homologous pairs. Each parent donates one chromosome to each of the 23 homologous pairs, which means half of an individual's chromosomes come from the female parent while the other half comes from the male parent. Homologous chromosomes are the same length and carry the same genes in the same location. Those genes could be different versions. Example would be, imagine that your mom has blue eyes, your dad has brown eyes. They're in the same place on each chromosome, but depending on which half you get determines your eye color. So we should keep in mind that there are two types of cells in general, right? We discussed the somatic cells, and that means that that's the diploid cells, right? And then when these split in half, we have the reproductive cells, which are called haploid cells. These haploid cells are called gametes and contain only one half of the number of chromosomes. If one somatic cell is fertilized by another, the resulting zygote would contain twice the number of chromosomes. A zygote carries a complete set of chromosomes, half from the female and half from the male. We should also know that this stage of splitting is called meiosis. Meiosis has the same main stages as mitosis, so we're not going to go through in such detail, but there are a little bit of differences. Meiosis happens in two stages, meiosis 1 and meiosis 2. The two cell divisions result in 4 haploid daughter cells. So during meiosis 1, the homologous chromosomes are separated. And you can see the chain happening here. In meiosis 2, the chromatids are separated. After the chromosomes are replicated, chromatids remain attached to the centromere. Also, homologous pairs, each consisting of 2 chromatids, remain close together. The 4 chromatids are called a tetrad, and the process is called synapsis. During synapsis, the arms of chromosomes in a homologous pair become intertwined. Pieces of the homologous chromosomes break off and switch places. The phenomenon is called crossing over. This increases genetic diversity, which results from sexual reproduction. Here you can see the breaking of the homologous chromosomes. And that, my friends, is mitosis and meiosis.